Hi, welcome to this lecture on environmental adaptation. This is the last video on this nutrition, feeding and digestion, and now we are going on the digestion and absorption. So let's have a full view of the digestion. What happens? When we have a cat that is eating the food, there is a mechanical cleaving, and so we can swallow some food particles. And after that, the cleaving is continued with chemical digestion. So these enzymes are working to get the molecules smaller. And then these nutrients are absorbed. And then undigested material is excreted. But there are also some exceptions on this general road. So in some cases, the digestion happens mostly inside the cells, so intracellular, in flatworms, some mollusks, etc. And then, as we already learned from those Hi, welcome to this lecture on environmental adaptation of animals. This is the last video on nutrition, feeding and digestion, and now we are going on the digestion and absorption part. Let's have a full view of what happens when we are eating, or the cat is eating. There is a food that is cleaved mechanical in the mouth and then swallowed as smaller food particles. The cleaving is continuing chemically in the stomach, in the intestine, to get the food particles in the small molecules that could be absorbed. And then these nutrients are absorbed in the digestion. And then this undigested material will be excreted. But there are also some exceptions in this general rule. In some cases, the digestion happens inside the cells, in flatworms and mollusks. And also that we have learned already in the previous video that actually the absorption can also happen even before the cleaving of the material. So in these ruminants, like these cows, they first had this fermentation, and part of that is producing B vitamins, and that is absorbed quite early. Okay, we have different segments in the digestion tract. It's divided always with foregut, midgut and hindgut. This foregut contains the digestive tract between the hindgut and intestine. So there is esophagus, stomach. In some cases, there can be crop, that is a stork chamber, or gizzard, that is a grinding chamber. And then we have midgut. Usually in vertebrates, it has some small diameters, and that's why it's called small intestine. And then we have hindgut, has a larger diameter and called large intestine. But the same parts are also found in insects and crustaceans. But as you can see, that over here the midgut is not so small diameter. So they must have different kind of absorption or digesting mechanisms. And in crustacean, there's in the stomach, there's teeth cutting the structures. And even there's a posterior stomach chamber that has cuticular bristle to strain the material. So it can have completely different kind of system. Or in the intestine, there's also these Malpighian tubules. So they got all the urea in the combined with the feces. So in general, what happens in different nutritions? We have carbohydrates, meaning sugars, we have proteins, nucleic acids and lipids. And this is something that you should already know. That in the mouth, we start cutting the carbohydrates. We have amylase in the salivary to get it a little bit smaller polysaccharides, but quite often, even in humans, this doesn't happen so much because we swallow the food quite fast. Then we have the stomach, where we have pepsin that is breaking proteins in small polypeptides. And then in the midgut, these polysaccharides are cut in smaller pieces with pancreatic excreted amylase. And the proteins, there is a trypsin and chymotrypsin that is getting these in smaller polypeptides, and then some other enzymes that are cutting it in amino acids. And in the midgut lumen, also the DNA and RNA is cutted by these pancreatic nucleases. But also in the lipids, we have these, it's formed in bile salts, and then we have pancreatic lipase that is cutting it in smaller pieces or modulated. But then in the epithelium of the midgut, it's also affecting. Not only that, okay, what's inside, but also the epithelia seems to be important. So we know that, okay, we have transporters, but also there are some enzymes that are cutting it in monosaccharides or amino acids, or some, we can cut these nucleotides in the nutritional sugars or phosphates, etc. And there can be variation among animals in these enzymes, like chitinase that is used for breaking the chitin. Okay, the layer, outer layer of the insects, and that's why rodents and bats have it. But we, or rabbits, we don't have the enzyme, so that's why we can't use the chitin. It would be a sugar. And also some other sugars we need to digest, and that's why some herbivores, they are using bacteria. Then the protein digestion, it depends on where it can happen. It can be happened with endopeptidases, so it can be cut in from the center of the protein, or in the end, so we have these exopeptidases. And also the digestion can happen in three different locations. We have intraluminar enzymes that are working there inside for example, the small intestine, and it's common in ours in humans. But then we have these membrane-associated enzymes that are on the surface of epithelia, but modulating these food particles in the lumen side. And then we can have also enzymes that are working inside the cells. And this is typical digestion in some clams. Okay, how this absorption in midgut is then done? Because absorption means different in different sorts of animals. So are they taking the nutritions in very small parts, or are they digesting inside the cells? And it can be used different kind of ways to transport stuff through the membrane. Simple diffusion for small particles, or those that are hydrophobic. Then we can have facilitated diffusion like ion channel. They can be active transport, and then can be some secondary active transport, so these co-transporters. And one of the things that is increasing this absorption is, of course, how much area you have around it. And that's why in the midgut you have these microvillis and etc. to increase the surface, so increase the area where this absorption can happen. And this we have already checked in some previous videos, but let's review it. So for sugars, we can have different kind of mechanisms. we have one protein that is affecting on facilitated diffusion for fructose. Then we have a secondary active transport for glucose and galactose. So although the fructose and glucose doesn't look so much different, they are going through a different kind of roads. And one need sodium gradient, one doesn't. And then on the other side, there is a uniporter that is facilitating this diffusion of these simple sugars in the circulation. And everything is worked with this sodium pump working as a workhorse to pump the sodium out from the cells, because otherwise these glucose sodium co-transporters would not work. Okay, then on the lipids. The fatty acids are going inside the lumen that is in these small balls that are rounded by bile salts. And these bile salts, when they are facing the microvilli, they will release the fatty acids inside the cells. And then these fatty acids are combined with glycerol to form some triacylglycerol. And then this glycerol is binded with xylomicrons. So binded with proteins to form some xylomicrons, because these triacylglycerols are very hydrophobic. So we need some transporting mechanism to get it in the circulation. Then what happens when we are eating? So it's regulated in the whole animal level. Because the digestion can operate only small amount of food per time. And when we swallow, it first happens with voluntary action, but then in the pharynx, and then involuntary as a wave of peristaltic muscles along the esophagus. And then there is a interplay between stomach and midgut that is affecting on the speed of the digestion. So the esophagus has two circular muscles or sphincters, and they relax when the food is coming from the mouth to the stomach. So they are preventing that, okay, the food is not going in the opposite direction. Then, when the food is inside the stomach, there are these G cells that are releasing a polypeptide, gastrin. And it's affecting on three places. On the cells that are secreting this acid, then the cells that are releasing peptidogen, so meaning pepsine, and then it's really affecting on the cells that is moving or contracting to move substance inside the stomach. And these are also regulated by neural activity. So if we see food or smell the food, it can affect on releasing pepsin in our stomach, etc. Okay, then when the food is going from the intestine to the midgut, there are three hormonal signals: secretin, cholecystokinin, and gastric inhibitory polypeptide. And they are all inhibiting the activity of the stomach, but also affecting that, okay, what happens in the liver, what happens in the pancreas. So it's quite understandable that, okay, how it works. Because we can only have in the midgut small piece of the food or small amount of the food at one time. So we must reduce the activity of the stomach when there is food near these secretion cells. And then when the food has gone a little bit further, then we can get the next section to digest it. But then some animals are not always eating. We can have long-lasting effects also. And pythons are a very good example of this. They deconstruct the gastrointestive tract between the meals because they are eating only once a week or once a month. And that's affect that, okay, the feeding triggers the synthesis of the epithelium with the transporters, etc. And it's affected by different genes. So about 6 hours after having the food, there is over 1,000 genes that are downregulated, over 1,300 genes that are upregulated, and it doesn't come without the need of energy, so the metabolic rate is 14 times more. And heart size is increased. Heart is larger. So it's very drastically affecting on the physiology or the morphology of this animal. And the digestion is also affected by environment. So we can have chronic changes that affected on our digestion by different environment, what kind of food we have. So this digestive absorption system can be deconstructed in new environment, or it can be modulated. So in humans, which have we need the sugar transporters to absorb the sugars in the circulation, and if our diet contains a lot of sugar, we have more of these transporters. So that means that, okay, if we are eating all the time sugar containing food, we get fattier with eating cake. Then the diet affects also on the enzyme secretion from pancreas. So in animals with having a fat-rich diet, they will release from the pancreas enzyme that is cutting more fat. If they are having a protein-rich diet, they will have these proteases, etc. But it also affects that in, for example, animals living in Finland in cold temperatures and being active, they have now a little bit larger gastrointestinal tract because they need more food to keep the body warm. Then this diet can affect on the gene transcription. So some genes are regulated by diet composition, and this is something so new that, okay, the consequences are not very clear. But at least in the diet that has fatty acids, it's affecting on the genes that are regulating the fatty acid metabolism. Then the nutrition can affect on the evolution. We already started this section by lactase intolerance. But it's also affected that, okay, in the animals that are eating the sucrose, they have the sucrase enzyme, because sucrose is not so common in the nature. And also the herbivores that are eating a lot of long plant sugars, and they have these microbes that are cutting the cellulose in the glucose. So they have also more glucose transporters. And then one thing that is affecting on the digestion is also development and these clocks. So the tadpoles are herbivores, but the adult frogs are carnivores. So they have a huge difference in their intestine during this development. Or then in regular mice, how they can have these able to cut the disaccharide with these disaccharide days, it's different at nighttime and the daytime. And this also affects that, okay, on how this digestion is working in the springtime or in the summertime, in the fall, and then in the wintertime. Like these crucian carbs that we have had as an example throughout this course, in the wintertime they are not eating anything. Even when we provide them any food, if the temperature is low, they don't eat. Thank you.